We ended our last video learning about the mitochondria and the chloroplasts. The mitochondria and the chloroplasts give us some clues about the evolutionary origins of the eukaryotic cell. So we are about to discuss something called the Endosymbiont Theory. This theory explains the origin of eukaryotic cells. I'm going to repeat myself in just a minute, but I wanted to introduce this term because I think sometimes students are blindsided by this big word and they don't learn the word, they don't understand what the word means, and then they brain dump this whole theory. So I want you to practice saying it, endosymbiont, and learn how to spell it, because when you do that, you're going to have a better chance of remembering what this theory is talking about. So let's talk about what this word means. Endo means inside. Symbiont comes from the word symbiosis, which some of you might be familiar with. This means a mutual relationship. So we are going to discuss the mutual relationship that occurred when one cell became part of another. In other words, two cells created a mutually beneficial relationship where one is inside the other. And the cells that we are talking about are eukaryotic cells. So we're talking about the origin of eukaryotic cells. And we can look at the mitochondria and chloroplasts for some information on how eukaryotic cells came about. Because if you remember a few slides ago, or at I guess the very beginning of the chapter 4 lecture, I told you that in our evolutionary tree, we have bacteria, and the bacterial line branched off and we got archaea. And then eventually archaea branched off. And we also get, so if there's our archaea there, we also get eukaryotes from that archaeal branch. So we are going to discuss the evolutionary origin of eukaryotes from that archaeal line. So how did eukaryotic cells evolve? Well, the short answer is they evolved from prokaryotic cells. So I already introduced the word endosymbiont. This means when one organism forms a mutually beneficial relationship inside another. So they both, in this case, cells, two prokaryotes, form a mutually beneficial relationship that is going to lead to eukaryotic cells. So the endosymbiont theory says that an early ancestor of eukaryotic cells, in other words, a prokaryote, engulfed, and we're going to ignore this for a minute, engulfed another cell and formed a mutually beneficial relationship. in which we now have eukaryotic cells with organelles. So let's go back and discuss what this other cell was. So an early ancestor of eukaryotic cells, a prokaryote, engulfed another cell. Now, this other cell had a really important characteristic, and that was that it could use oxygen. So an early prokaryotic cell engulfed another cell that could use oxygen. And what that cell was doing was using oxygen to convert energy. So if we look at this whole definition, our endosymbiont theory says that an early ancestor of eukaryotic cells, a prokaryote, engulfed another cell that could use oxygen to convert energy into a usable form in the cell. Now, it's important that this says non-photosynthetic. We're going to get to that in just a minute. So the endosymbiont theory tells us that the origin of the mitochondria in eukaryotic cells came from one prokaryotic cell engulfing another. Now, that's how cells eat sometimes. They engulf whatever they're going to eat. They surround it with their plasma membrane and pull it in. We're going to learn about that in chapter 5. And normally when you eat something, you digest it. A cell's the same way. If it pulls in a food particle, it's going to eat it. But in this mutually beneficial relationship, that energy using cell was not broken down. It was not digested, and it became a part of the other cell. So what we have here is one cell, a prokaryote. We got some DNA in there, but we don't have any other organelles. And that cell is going to consume another smaller prokaryotic cell. And what we're going to get is that large cell with the smaller cell inside, and that guy is going to use oxygen to convert energy in the cell. Now I want you to notice something. That little prokaryote that was digested still has its own DNA, and that's important. That's going to help us sort of back up this theory with some evidence. So two cells formed the symbiotic relationship, producing an endosymbiont that was an ancestral prokaryotic cell. I got ahead of myself a little bit. So in the next step, we have our eukaryotic cell that is picked up this little precursor mitochondria. We're going to start calling it the mitochondria because it's going to evolve into the mitochondria. So these guys go along happily as eukaryotes, and they are eventually going to lead to our animal cells. But one of those eukaryotic cells decides to take its own pathway. And on this pathway, and it probably happened multiple times, but on this pathway, that cell that has picked up what will be our mitochondria decides to eat something else. And this guy is going to eat another prokaryote that can use sunlight energy to convert it into usable energy for the cell. So this guy can perform photosynthesis. So our big cell is going to eat our little precursor chloroplast. So now we have another symbiotic relationship. Where this cell now has a mitochondria and a chloroplast. And that is going to be our plant cell ancestor. So this endosymbiont theory gives us an evolutionary pathway for the development of our two major eukaryotic cell lines. One that contains only mitochondria. Dry that guy up here, the animal cell. And one that contains both mitochondria and chloroplast. So that explains the origin of our non-endomembrane system organelles. Our textbook doesn't really go into the evolutionary origin of some of the other organelles, basically all of them that fall under the endomembrane system. If you're interested in that, come chat with me. It's very simple, but we're not going to go into it here because our book does not. So here's a diagram from our textbook of the endosymbiont theory. It's a little bit more detailed than mine. Up here we've got our eukaryotic ancestor. In this image, they already have the nucleus and they already have some of the endomembrane system. So this is not a true prokaryote at this time. It's a prokaryote that has already begun the evolutionary steps towards being a eukaryote. But we've got this diagram that shows engulfing of the oxygen using non-photosynthetic prokaryote, this guy, which is going to evolve into the mitochondria. Now, some of those guys are going to continue on to be animals. I think this fungi is actually a typo here. Fungi are generally more closely related to plants, but that's okay. It's not a huge deal for the moment. And then some of those cells are going to move on to engulf a chloroplast and become the ancestral cells to our plants and other photosynthetic cell types. I wanted to add one thing here, and that is our lines of evidence for the endosymbiont theory. Now, the first one has to do with the structure of those mitochondria and chloroplasts. Remember when we talked about the structure, we said that both the mitochondria and the chloroplasts have double membranes. Now those double membranes are very, very similar to the double membrane plasma membrane on the outside of the cell, which suggests that they are originally independent cells. The other line of evidence for the endosymbiont theory is that both the mitochondria and the chloroplast contain their own DNA. Now, they'd only have their own DNA if they were originally an independent cell. Today, the mitochondrial DNA and the chloroplast DNA only contain instructions for building more chloroplasts and more mitochondria and for mitochondrial and chloroplast function. But in either way, it still has its own DNA. It is small plasmid DNA, which are little tiny circular pieces of DNA, much, much smaller than chromosomal DNA, but does encode its own function. So those cells actually, those organelles actually express their own genes independent of the greater cell, further lending support to the endosymbiont theory. So our last section is kind of structures that surround and support our cell. So in chapter 5, we'll talk about the plasma membrane. More than you ever care to talk about plasma membranes, more than you will ever talk about plasma membranes in your life. So we won't go into too much detail there. But the plasma membrane is essentially the structure that holds all the cellular content. So it surrounds the cell, it isolates the cell from the environment, and it regulates what moves in and out of the cell. We'll talk about the properties of that in chapter 5. Generally, it is a double layer of phospholipids. So between now and next week, you'll probably want to review what a phospholipid is, maybe what a lipid is. We talked about phospholipids in chapter 3. Inside the cell, underneath and within the plasma membrane, is the cytoskeleton. So the cytoskeleton is almost like scaffolding for the cell. It's a network of cellular fibers that spread throughout the cell, and they do a few different things. The first thing that they do is that they support the cell. They provide structure for the cell. And the cytoplasm is made up of the largest type of cytoskeleton elements called microtubules. And there are some other elements. We're not going to focus on them too much. We're going to come back to one of the functions of microtubules later. Those are just the biggest ones. If you look at this image here, these green structures and the red structures, those are all different types of cytoskeleton elements that are giving structure to the cell. They support the cell and support the cell membrane. The cytoskeleton also organizes the structures within the cell, so they'll provide support for the organelles, and also can act as, again, scaffolding or support structure that different processes can take place on or move along. One of the important functions of the cytoskeleton is movement. So their cytoskeleton elements are involved in cell motility, which means movement of the entire cell. This happens by structures called cilia or flagella. So cilia are little almost hair-like structures on the outside of a cell, and they beat like oars. They kind of move back and forth together in a wave pattern, and they'll do things like move fluid along the outside of the cell. And then some cells might have, not necessarily in addition to, most of them don't have both, might have flagella. So a flagella is a single long structure that is important in basically like propelling the cell through the environment. A good example of this that you all would be familiar with are sperm. So cilia can occur if it is a cell that is stationary and we're moving things around the cell, but cilia or flagella can also move the cell through its environment. The cytoskeleton also helps movement within the cell, and this is done by using motor proteins. We're going to come back to those, but when we talk about movement in the cell, we are talking about essentially moving products through the cell. So a little protein packet called a vesicle can actually essentially walk along the cytoskeleton, and that's how we move products within the cell. Cytoskeleton is also really important in moving chromosomes, and we will come back to that when we talk about cell division, when we start sorting and partitioning our chromosomes. Outside of the cell membrane, in plants, fungi, and many bacteria, we have cell walls. So the cell wall is a structure that occurs outside of the plasma membrane. So if you look at this picture here, the plasma membrane is this orange line. It's really small. The cell wall occurs on the outside of that. Plants, fungi, and bacteria all have different types of fibers that make up their cell wall. We talked a little bit about cellulose in chapter 3. That is the fiber that makes up the plant cell wall. And the cell wall gives support and structure for the plant to stand up straight. Just basically gives a rigid structure outside of the plasma membrane. You can also have communication between cells through the cell wall. So there's a whole chapter on cell communication. A whole section on cell communication we're not going to go through, but they're basically gaps in the cell wall that allow cells to communicate with one another. Animal cells don't have a cell wall, but they do have a rigid, semi-rigid outer structure called the extracellular matrix. It is a set of fibers that provide support outside of the cell. So it's these purple fibers and these green fibers that attach to proteins that are embedded on the outside of the plasma membrane. And along with the cytoskeleton on the inside, they give sort of an outside support structure for the cell. You can also have communication between animal cells, just like in plant cells. Again, we're going to skip that section in this chapter.